The next talk will be delivered by Dr. Chin-Hoe, and let me take this opportunity to briefly introduce our speaker. Dr. Chin-Hoe obtained his PhD in pharmaceutical chemistry from University of Science, Malaysia. He joined Brooklyn, Malaysia as an NMR application scientist. He supports Southeast Asia region and provides the assistance to the customers, especially who are new in the natural product plant metabolomic fields. His expertise is with different latest software solutions, both for pure, compound, and mixture to drive many possible NMR applications into the easier and better way. He is part of the NMR method and application development, and his NMR application interest includes metabolomics, quantitative NMR, and fast NMR techniques. So today he will give a talk on the title, A Quick Update to SARS-CoV-2 NMR Metabolomics Research. Ladies and gentlemen, please join me in welcoming Dr. Chin-Hoe.
Thank you, Dr. Jutara, for inviting me to present this talk. I will share my screen. So today I will talk about the quick update on SARS-CoV-2 on the NMR metabolomics research. So as we know that the SARS-CoV-2 virus, severe acute respiratory syndrome, coronavirus 2, is the virus that caused the COVID-19 disease. So when we take a look into the structure of this SARS-CoV-2 virus, so it belongs to the beta coronavirus genus. It is a single-stranded positive sense RNA with a nucleocapsid and protein. And the genetic contents is encapsulated by the viral envelopes. So when you look into this, the single-strand RNA, so this is the membrane that cover or to coat all the RNA. And beside this structure, so the virus also contains the spike glycoprotein, which is on the surface, membrane protein, and then also the envelope protein, which all three are embedded on the surface of the membrane. So when we look into the research that related to the SARS-CoV-2 virus, so we have to think about the safety. So there are 4 different biosafety level in general. So biosafety level 1, 2, 3, and 4. So different biosafety level have their own requirements. So this is a biosafety level number 1, biosafety level number 2, biosafety level number 3, and biosafety level number 4. So when you look into the biosafety requirement that related to the SARS-CoV-2 research, so for if you are handling the sample which are related to the culture or the specimen, so usually you require the biosafety level number 3. But if you are working on the diagnostic routine, okay, or the diagnostic testing, so you only need the biosafety level 2 for the requirement. So why we only need the biosafety level 2 laboratory? Because in preparation of the sample, so usually the microbiologists will try to lyse the bacteria with the buffer, or they can perform the high temperature to destroy or to inactivate the virus. So for example, 56 degrees Celsius for 30 minutes. So this will make the sample safe to human or safe to the worker. So in general, for the SARS-CoV-2 scientific analysis using the NMR scientific research, so there are 2 main groups of the research directions. One is related to the fundamental research, which is the study of the RNA or the structure, secondary structure RNA, or the protein that related to the SARS-CoV-2 virus. So another important direction, which is we are going to talk about this today, is a metabolomics approach that is usually can be related to the future diagnostic. So for the metabolomics that related to the SARS-CoV-2 research. So we only need a laboratory with the biosafety level 2. So when you look into the most of the publications that you can find from the literature review, so the sample that collected from related to the SARS-CoV-2 research are mostly plasma, and some of the publications they are study on the serum. So which is the blood, okay, yeah, the blood. So this is the example of the plasma NMR result, okay, and this is another example, is a serum NMR that come from the COVID-19 patients, and another lower trace. This is from the healthy control, which is a pre-COVID. Yeah. So the fingerprints are pretty complex, but very informative. So as you know that for the SARS-CoV-2 research that using the NMR, so you only need the biosafety level 2. So the next question is we're going to ask is it necessary for us to destroy the sample by using the heat treatment to inactivate the sample? So there are two reasons that are related to this answer. Okay. So one is when you look into the when we look into the sample itself. So for example, in these publications, so it's using, okay, they are using the plasma and the serum. So this plasma and serum, the sample amount is only 350 microliter of the plasma or serum, and this sample is mixed with the buffer. And then from this total, so they only take out 600 microliter of the mixture that put into the NMR tube for the NMR experiment. So it means that in terms of the volume itself is not very much. And we also know that for this COVID-19 disease is transmitted through the droplets. Okay, so as long as you are using the protecting glove, okay, the handling the plasma do not need very high highly security. So means that highly biosafety means that we can still handle this with just the biosafety level number 2. So another reason why we do not need to heat up we do not need to apply the heat treatment in order to deactivate the sample is because when you perform the heating, so most probably you will introduce the unpredictable result. So because as you can see from this PCS score plot of the serum sample, so this heating of the sample, yeah, heat treatment of the sample will introduce the non-uniform alterations in the chemical composition. So most of the sample, the behavior will be changing. And you will introduce the false positive discriminator or artifactual pseudo biomarker. So this is the second reason why when we work with this SARS-CoV-2 sample using NMR metabolomic approach, so we do not need to apply the heat treatment. So how can we positively identify the COVID-19 patients? So there are two ways that we can test or we can identify whether the patient has been, the people have been infected or not. One is through the PCR. So this PCR is through the direct detections of the RNA from the virus. This is one way. So if the virus entered the human body, and it will trigger up our human immune response. So we also can consider the antibody test for the patients, because as the body defense build up, so the body will produce the antibody. So we can also use the ELISA test to detect the antibody through this serological test, yeah, or the antibody test or immunoassay test. So one of the important research group from Australian National Phenome Center is led by the Professor Jeremy Nicholson and Professor Elaine Holmes. They did a number of research that related to the SARS-CoV-2 using NMR study. So here, this is one of the recent publications from the Professor Jeremy Nicholson on SARS-CoV-2 study based on the metabolomic approach. So using NMR and MS as an integrated technique, so the initial result actually indicates that there is a high promise of using this approach for developing novel SARS-CoV-2 diagnostic tests based on the terms called the phenol conversions. So this is a definition. The phenol conversion is defined as the systemic shift in the multiple molecular phenotypic properties from the healthy to a characteristic activated or pathophysiological states in response to an infectious or an pathophysiological challenge. So according to these publications and from this study, so the plasma sample were collected from 17 adults who presented the COVID-19 disease symptoms and subsequently tested positive for SARS-CoV-2 infections from upper and lower respiratory tract swabbed by RT-PCR. And 25, the healthy control, they are recruited from the population who did not and had not shown any COVID-19 disease symptom. And they were serologically tested negative with respect to IgA and IgG antibody. So here, let us look at the results. So this is the untargeted principal component analysis PCA score plot on plasma obtained from COVID-19 patients and the healthy control. So as you can see here, the healthy control are highlighted in the blue, and then the patient are highlighted in the red. So the positive patients here, they are showing some kind of a clustering. Okay, yeah. So even though on this this is a statistical routine, PCR already can show the differentiation power. So when you look into the more advanced statistical analysis, such as OPLS-DA, so this is the OPLS-EDA that performed on the same plasma data obtained from the combined approach, which is done by combining the NMR and MS. So, and here, as you can see here, that the OPLS-DA show the very similar patterns to the PCA plots. So the red triangle, these are from the positive patients group, and the blue triangle, these are from the healthy control. And in addition to that, the two sample, which is highlighted in the greens, okay, were collected from the individual A and B. So this individual and A and B, okay, they are initially tested the SARS-CoV-2 RNA positive, okay, but have already recovered from the disease when the sample is collected. So both individuals showing the coordinate which is close to or near to or within the healthy group regions. So this supervised OPLS-DA methods actually give a very clear differentiations between the COVID-19 patients group and the healthy group. And another example to show, which is the C. Okay. So interestingly, the sample from the C, it shows that this is the patients with the mild symptomatic and who was unaware that they suffer from the disease, but subsequently tested that they found to be antibody positive for the SARS-CoV-2 IgA and IgG positive. So this concludes that actually the NMR can give the correct answer, even though this patient is with the very mild symptomatic. So when you look into the result, so now we can look into the how can we differentiate between the healthy and the COVID-19 positive patients sample from the NMR result. So the current COVID-19 study shows that for the COVID-19 the patients or the SARS-CoV-2 patient, they demonstrate an abnormal lipoprotein metabolite signature, which is consistent with the coronary artery disease. And the result for the related to this lipoprotein profile, so they give the low total and the high density lipoprotein, apolipoprotein A1, this is a very low concentration in the high density lipoprotein triglyceride and the high LDL, low density lipoprotein and the very low density lipoprotein triglyceride. So this is the result, and this lipoprotein related parameter play a substantial role in early biomarker identification, and this is very exclusive to NMR. So besides that, the discriminatory biomarkers also can come from the amino acid category as shown in this table. Yeah, so which is also important to explain the COVID-19 mechanism. So beside the lipoprotein profile, so the COVID-19 patient also give the very significant and very characteristic in terms of the amino acid profile and also the sugar. Okay. So as we know that the COVID-19, the disease affected the human in a very complex way. Infections of this SARS-CoV-2 may lead to the multiple organ failure, especially in the severe case. Hence, the coronavirus 2 is a critically ill patients. So they will cause a complex pattern of a disturbance, especially related to the systemic metabolism. So it means that it will affecting the whole body instead of just affecting the lung only. So when you look into this test, so for the RT-PCR test, so yeah, in general, yeah, the PCR tests are likely positive in the first week of infection. So this is here, yeah. So if you after the third week of infection, so the PCR test is become negative. So this is the limitations of the RT-PCR. And when you look into this antibody test, so the antibody test is only active on the weeks of 2 and onwards, start from the week 2, week 3 onwards. Okay, but when you look into the approach that's suggested by this research group, there is a metabolic conversion. Actually, is a very powerful tools that it can allow us to cover all the COVID-19 disease phases. So it allows us to detect this metabolic metabolites, especially in the earlier stage, especially related to the incubation periods. And then it also allows us to detect when the patient is severely affected by this disease. Okay, and you also can use this phenol conversions during the recovery phase, so we can perform the long-term disease risk predictions. Okay, yeah, this is also possible with this approach, which is a phenol conversions. So then another research paper that published in this group by this CIC Biogun. So in this in this paper, so they try to analyze the NMR metabolomics in the larger cohort. So in this study, so they recruit 263 COVID-19 patients, and then they take the serum, and then they compare with the 280 individual from the pre-COVID during the pre-COVID time, the serum, and then they compare the statistical analysis. So they want to see whether it can show the clear manifestations of the disease. So here, this is the PCA result, the score plots of the PCA from the serum metabolite NMR, and then this is the PCA score plot from the lipoprotein subclass data. So as you can see here, that both PCA results show the very reasonable separations between the COVID and the pre-COVID cohort. So they are showing the very good grouping in the PCA. So in the OPRS, whereas in the OPRS-DA, so you also can see the very clear groups between the healthy, or we call the pre-COVID, and the COVID patients. So these differences is observed by the unsupervised analysis, such as the PCAs that I mentioned previously, was confirmed by the OPRS-DA model here. And actually, in this result, it showed a very reasonable degree of clustering. Okay, yeah. So another paper that come from the Australian Phenome Center by Professor Jeremy Dickerson's group. Okay, so in this paper, so they will try to investigate the systemic metabolic effect of the SARS-CoV-2 infection. So we know that the virus will not affect the particular organ, but it affects the whole system metabolomics. Okay, systemic metabolomic effect. So this is one of the results to show the overall group. Okay, yeah. So we will have this healthy control group. Yeah. So this and then this is from the healthy control group. This is the patients group. So this is a PCA from the 1D result from the obtained from the 600 megahertz animal instrument. So consider the PCA a model of the 1D spectra data set. So the healthy control are the healthy control, which is indicated as in blue. So they are clustering on the left side of the score plots, and then for the patient or the positive COVID-19 positive patients, they are clustering to the right of the diagram. Yeah. So then when the patients are tested the negative for SARS-CoV-2, so they were mainly non-hospital. So this is the light gray color. Okay, they are non-hospitalized, but this is from the COVID negative result, and tend to cluster with the healthy group because they are healthy. Okay, and then while the patients are hospitalized for other respiratory conditions, so which is highlighted in the dark gray. Okay, yeah. So this is the other than the COVID. So some of them are fall in the patients group. Some of them are in betweens. Okay, yeah. So the 7 out of 34 participants originally recruited as a healthy control. So this is indicated by green symbol. They were found to have the high IgA and IgG level. So suggesting that, so as you can see here, the green color is here. Yeah, all these are the patients that they give the positive test in the immunoassay. Okay, immunoassay IgG and IgA. So suggesting that either this individual have previously been unknowingly infected, but were asymptomatic or mildly affected, or that they may have some cross-reactivity with some other coronaviruses. So this individual met in between the SARS-CoV-2 positive, which is in the red, and the healthy control, which is in the blue, in between. So with one individual mapping within the SARS-CoV-2 positive cluster, which is identified by this circle, yeah, coordinate. So this will give this particular people or patients, they give the positive mapping within this SARS-CoV-2 cluster. So it means that for the antibody test, so this is positive, but for the RT-PCR is not detected. Okay, yeah, but this is fall under this patients group, yeah, positive cluster. So the seropositive but not RT-PCR confirmed sample lie at the boundary between the SARS-CoV-2 positive and the healthy group indicates that there are some metabolic similarity between the SARS-CoV-2 positive group and may represent the participant who have been exposed to the virus. So it means that actually the patients already exposed to virus, but in the test of the RT-PCR, they are not detected. Okay. So this is the another result from the same data, but plot in the OPSEA. So again, as you can see here, the very clear groups, okay, differentiate between the healthy and the SARS-CoV-2 positive patients. So this is another result from the lipoprotein. So just now is the full spectrum, but now they are looking at the lipoprotein parameter. So as you can see here, that the most of the sample from the healthy participant, which is the blue. So including those from the healthy group who had tested positive of the IgG. So here, this is the positive IgG. So they are grouped on the left-hand side of the graph. And then for the SARS-CoV-2 positive patients, so which they tend to gather or they cluster at the right-hand side of the graph. So when you look into this lipoprotein, actually the profile is the result is very similar to the full spectrum. So this is one of the sample that we collected. They collected the same from the same patient, but at the different time point. So here, this is the lipoprotein profile over 5 collection time point for the same patient. So as you can see here, yeah. So this is the time point 1, time point 2. So it's from this plot. So they have the red star. Okay. So this red star represents the time point where the participants are tested PCR positive, and then the cyan star. Okay. Yeah, which is number 5 here. So this is already tested negative. So within this SARS-CoV-2 positive group, because this is the positive group patients, yeah. So as shown in this slide, so represent multiple sample and multiple sample measurement in terms of the NMR, plasma and the plasma. So they show them, actually it showed some kind of a metabolic trajectory. So with this, you also can see the progressions of the infections. So longitudinal trajectory analysis were performed on one of the patients, which is here, and as indicated here. The biochemical profile met within the SARS-CoV-2 positive space. So this is within the red color zone, okay, which is in the graph, okay, of the PCA. So so when the patients when the patient is tested at the time point number 5, so the patient shows that the RT-PCR shows that this is already the negative, okay, negative. But however, when you look at the profile, so this profile is not looked to the normal, okay. The normal, it should be in this particular region. So this is already the RT-PCR already show the negative, but from here the hospital usually will discharge the patient. Yeah. So this raises an interesting and important clinical point that the recovery of these particular patients, okay, from the COVID-19 is usually accessed by or based on the respiratory syndrome. However, a patient cannot be said to be truly recovered unless they are also biochemically normal and free from some of the more complex and less visually obvious systematic effect of the disease. So it means that if these particular patients, the NMR data can go to this particular blue region, then we can say that the patient is fully recovered. Yeah. So from the trajectory, so it shows that we able to here we diagnose the patient at the time point number 1, and then after that we measure the time point number 5 for the RT-PCR. So we don't have any testing in between. So it means that actually we miss some of the time point. So this observation suggests that the SARS-CoV-2 infections leave the residual metabolic signature with and yet undetermined, okay, time of resolution. So this type of information could be clinically useful in determining the rates of recovery of patients, or indeed incomplete recovery with respect to the new onset comorbidities. So thus, these patients in the respiratory recovery phase and testing virus free was still metabolically highly abnormal, because this is still under under this red group, okay, and which indicates that the new role of NMR metabolomics in accessing the full systemic recovery. So it means that the NMR actually can be a useful technique to study whether the patient is fully recovered from this biochemical biochemical result. So this is the OPSDA from the same result. Yeah. So as you know that we will try to focus a little bit on the more severe case, okay, yeah, because this is critical level. So usually which involve the acute cardiac injury or multiple organ failure. So this is the more severe case. So when you look into the immunopathology of the COVID-19, so usually for those people who suffer from the severe COVID-19 disease, so they have the key characteristics, which is they show some kind of the cytokine, the increased productions of cytokine. So there are many types of cytokines that present in our human body. So this is the overall cytokines that from this particular review paper. And similar from another review paper, so they also talk about the cytokine storm that that appear in the severely affected or the patient that had admitted to ICU. So there are many cytokine individual cytokine involved, okay, or the chemokine involved. Yeah. So when you look into if the patient suffer from the severe clinical features, so usually you associated with the multiple organ dysfunction or the multiple organ failure. So this will always link to the cytokine storm. So this what is a cytokine? So when you look into the cytokines are low molecular weight proteins that act as a chemical signal in the immune response to pathogen invasion. So the productions of various cytokines in response to an evading pathogen such as a virus contributes to the host organism ability to eliminate the pathogens. So this is the functions of the cytokine. So actually it's a protein, okay, it's low molecular weight protein. So in this paper, okay, so the group also tried to look into how the NMR data can be linked to the cytokine. So in this result, so they try to correlate, okay, the cytokine result, okay, with the NMR-based lipoprotein from the main fraction parameter. So from the lipoprotein, this is obtained from the NMR spectroscopy data. So the lipoprotein parameter shows a strong correlations with the multiple cytokine. So you have many cytokines that try to relate, and actually there are 4 main clusters from the cytokine lipoprotein correlation pattern were observed. So for example, here they give the label C1, cluster 1, cluster 2, cluster 3, and cluster 4. So in particularly, so they are looking at the 2 more, due to the time, so I only go to the 2 important cytokine, IP10 and the rentis. Okay, so in the next slide, yeah. So this is also the cytokine result related to the NMR-based lipoprotein subfraction. So previous slide is the main fraction, now it's a subfraction. So looking at the same cytokine, so this IP10 and the rentis, okay, so this belongs to the subcluster C3. So these show the strongest associations with all the cytokine with the lipoprotein panel. So it means that it can link to the lipoprotein. So for example, it can link to the L1 to L4 LDL subfraction and anti-correlate with the HDL, VLDL, and the L6 LDL parameter. So actually this particular paper, it shows very good, beautiful merging the combined informations from the metabolomics point of view and the proteomics point of view. Yeah. So we can study this COVID-19 not just based on the metabolomics, but also we can combine the informations from the proteomics. So as you know that the NMR, the plasma example, okay, especially the plasma of the serum. So the lipoprotein is important. So the Bruker also, we have the platform that able to analyze this the lipoprotein. So we call the BI-LISA lipoprotein subclass analysis. So in this BI-LISA subclass analysis, so we able to analyze the spectrum. So after you have your NMR data that collected at your spectrometer, so this data can be sent to our server at located at the data analysis server at Germany. So from this server, so the server will perform the NMR spectrum analysis, and then you will have the reports. So from this report, then you will know whether which particular group or the main group or the subclass of the lipoprotein have been changed by this COVID-19 disease. Yeah. So the BI-LISA actually is one of the important and useful tools, okay, especially to address the COVID-19 disease. So here, this is the international network, okay, from the International Phenome Center currently. So these are the members that in the group. So here, yeah, I want to draw some make some conclusions. So NMR actually is a robust and highly reproducible spectroscopy method. And it allow us to integrate the spectral data from all over the groups into the post-processings like the statistical analysis and quantification. So it means that you can share your data amongst the lab as long as you follow the SOP. And the International Phenome Center Network uses this approach for the COVID-19 research. So because we need to collect many data, and then we need to share the data, and then we can combine the data as well. So NMR can support the COVID-19 analysis for acute infections, severity, and also the post-COVID syndrome. So on this platform, there's an efficient enabling tool that allows us to generate a large number of parameters. For example, the lipoprotein parameter from one measurement. So you can perform the targeted analysis and the non-targeted analysis as well. So the lipoprotein subclass analysis is important to generate about 115 lipoprotein related parameters in one measurement under full automations. Clinical relevance was shown. So if you are interested to understand more how NMR can be applied to the COVID-19 research in general, so you are welcome to go into this particular link, and you can download this video, and then you can watch this video on demand. So with this, I finish up my presentations. Thank you very much for your attention.
Thank you very much, Chin-Hoe. And maybe we are okay for just one question for now because we are falling behind our schedule. So if you have any question, you can speak through the microphone, or if you are more convenient for the chat box, please do so.
I think there's a question in the chat box. There's 2 questions.
Oh yeah, let me enlarge the chat box. I can see, yeah. So one is from Kun Vivek. So the question is, have you tried to find any difference between the fully recovered and long COVID or chronic COVID patients suffering subjects in terms of lipoprotein data, if any?
Okay, so actually the research is still ongoing. So until now, our collaborators still we try to collect some other data after the several months of this COVID disease gone. Yeah. So this this is the still ongoing research. Yeah.
All right, and probably just the second and last question for now. Can we select a few metabolites from this study to develop a more easy and cost-effective detection method in the mild for COVID-19 diet may not be suitable for all hospitals?
Okay, so actually for the NMR result, so it showed a very specific on the lipoprotein profile. Yeah, so as long as you have the lipoprotein profile as the biomarker, then you can already analyze, or you can use it as a diagnostic, yeah, for the disease stage, acute stage, and also for the recovery stage. Yeah.
All right, thank you, Chin-Hoe. And if any of the participants have any more questions, please feel free to drop your questions in the chat box, and we will gather all the questions into our panel discussion at the end of our webinar today. So hand over to Dr. Saptar, please.